Welcome to Topic 7. Today we're going to be looking at molecular biology. As the name suggests, molecular biology is the study of the structure of molecules and their interactions inside living cells. We're going to be focusing on the genetic material of the cell, DNA and RNA, looking at how it's replicated, looking at its structure, and talking about how the information contained within those molecules is decoded to manufacture protein. I'm hopeful that much of this will be review, that this is stuff you've seen in high school, but if you don't remember it, that's okay. This will be a quick refresher, and then we'll talk about how molecular biology can be used to do all sorts of cool things like genetic engineering. Now, this is important stuff. This is going to be critical for when we talk about viruses. So if this stuff isn't making sense to you, make sure you look it over and make sure that it does make sense to you before we get to our next topic. Genetics, as we'll see, is based upon an underlying principle known as the central dogma of molecular biology. And it's not nearly as frightening as it sounds. It simply states that cells contain DNA as their genetic material. DNA contains information, and that information is passed down from one generation to the next. To do that, of course, DNA has to be replicated. So before a cell divides, we have to have two copies of that information. DNA serves as a template to make RNA, and then RNA is used by ribosomes to make proteins. So DNA is transcribed into RNA, and then it's translated by a ribosome to make protein. The information that's contained within DNA and RNA is information needed to arrange amino acids in a particular sequence. So DNA is our genetic or hereditary material, and protein, remember, is the functional material of the cell. So proteins are enzymes that can manufacture lipids and carbohydrates, enzymes that can do all sorts of things. Proteins are channels. There are motor proteins, etc. So proteins do all the work. The DNA is simply the information. Within the DNA, we have discrete sequences that code for particular proteins, and those discrete sequences are known as genes. Now, a lot of people will tell you that the nucleus of a eukaryotic cell is the brain of the cell. I don't really like that analogy. I prefer the analogy of a library. The nucleus is the library, and it contains bookcases filled with books. Each bookcase is a chromosome. And on a particular bookcase, we're going to have a particular set of books. Each of those books is a gene, and each one of those books contains the blueprints for one particular protein. Bacteria, of course, don't have a nucleus. Now, they still have genetic information. They have DNA, of course, but they have it in one great big circle. And that's not contained in a separate compartment. So the first step, transcription, where we take DNA and use that to make messenger RNA, that would occur within the nucleus in a eukaryotic cell. And then the messenger RNA that resulted would have to move out into the cytosol and find a ribosome. Well, in bacteria cells, that messenger RNA gets dumped into the cytosol immediately, and then we have ribosomes that are nearby, in contact with the DNA in some cases, that can then translate it immediately. This speeds up the process quite a bit. In fact, as transcription is occurring before the messenger RNA has even detached from the DNA, it can already be in the process of translation. And this is one of the reasons that bacteria can divide and do stuff really, really quickly. Alrighty then, let's start by taking a look at the structure of DNA. The structure was unraveled, so to speak, by Watson and Crick back in 1953. Now, they didn't do their own empirical lab work. What they did was they looked at the findings of many other scientists. They compiled those findings and came up with a working model of DNA. You can see them doing that in this classic photograph. So they have scrounged the lab for various bits of equipment, retort stands, etc., to make a working model. They published a groundbreaking paper that was actually only 1 page long, you're seeing half of it right there, that contained their model. And this was a really big deal, of course. If you go to Cambridge, you can still see the stained glass windows that were erected in their honor. Now, DNA was discovered quite a bit earlier, in 1869 in fact. And we knew quite a bit about the components of nucleic acids, RNA and DNA, before Watson and Crick came along. We just didn't know how those bits and pieces were stuck together. Nucleic acids are built using nucleotides, and each nucleotide has 3 parts. It has a phosphate group, a sugar, and a nitrogenous base. The nitrogenous bases are so named because they contain quite a bit of nitrogen, as you can see. And the nitrogenous bases come in 5 varieties. We have one group known as the purines, and they're wider. They contain 2 organic rings. And then we have a second group known as the pyrimidines that are narrower, consisting just of a single ring. We have adenine, guanine, thymine, and cytosine in DNA. In RNA, we have the same, except thymine is replaced by uracil. Let's take a look at the sugars that are found in nucleic acids, and we'll start with the sugar that's found in RNA. That sugar is known as ribose, and RNA stands for ribonucleic acid, so the name makes sense. Note that we have 5 carbons, and they're numbered 1 through 5. Actually, they're numbered 1' through 5'. The first 4 carbons, together with an oxygen, form a pentagon. Note that on the 2' carbon, we have a hydroxyl group, an OH. Now, if we take ribose and we remove the oxygen from that location, we get deoxyribose. D means without. Oxy refers to oxygen, so we have removed 1 oxygen from that sugar, and that's the only thing we've done to change it to deoxyribose. Technically, if you want to be 100% correct, it's 2-deoxyribose. The 2 tells you that the oxygen is missing from that second carbon. Nucleotides are linked together in a chain using what's called a condensation or dehydration synthesis reaction. And we talked about these in Biology 111. Remember that all polymers in living cells are made through this reaction where 2 monomers come together and a water molecule is drawn off, hence the condensation or dehydration. So we have nucleotides that are coming into this growing chain. Those nucleotides will have 3 phosphates attached to them. That's not shown in this diagram. 2 of the phosphates would be removed, leaving 1. That 1 phosphate, so we're looking at the nucleotide on the bottom left here, has an exposed hydroxyl group. And it's going to interact with the exposed hydroxyl group on the 3' carbon of the existing strand. The hydroxyl group from the phosphate and the hydrogen from that 3' carbon are going to combine to give us our water, and we have a bond forming. The linkage between the nucleotides is known as a phosphodiester linkage. And this is what happens in both RNA and DNA to build a strand of nucleotides. RNA consists of a single chain of nucleotides. That chain forms a strand, and that strand is twisted. It's a spiral or helix. DNA consists of 2 helical strands of nucleotides twisted around each other. And that's why Watson and Crick referred to this molecule as the double helix. What you're seeing here are 3 different representations of this molecule. Let's take a look at the one in the middle first. In a DNA molecule, we have 2 strands, and each one of those strands has a backbone. The backbone is made up of alternating phosphates and sugars. So we have a backbone here on the left, and we have another backbone here on the right. Now, if you look at the diagram on the left, that backbone is represented by blue ribbons. So we have a backbone here, and we have another backbone over here. The two backbones are going to be held together by our nitrogenous bases. The nitrogenous bases always pair up in a very specific way. So adenine, A, binds to thymine, T. Cytosine, C, binds to guanine, G. Note that we have 2 hydrogen bonds that hold A and T together, and we have 3 hydrogen bonds that hold C and G together. So the nucleotides will always pair up in a way where they get the maximal number of hydrogen bonds. In fact, if you have a sequence of DNA that has a lot of G's and C's, there will be more hydrogen bonds holding the 2 strands together, and you'll have a stronger molecule. The other thing to note when we look at the backbones is that they're arranged in opposite directions. Take a look at the position of the oxygen on the pentose sugar. It's different. And that's because of the way that strands have to be built. As we talked about before, new nucleotides have to be added to the 3' hydroxyl. On the right, you're seeing a space-filling model of the DNA molecule. And note again that we have 2 strands that are wrapped around each other. They're twisted around each other to form this double helix. The 2 strands of nucleotides that are found in a DNA molecule are said to be complementary. And what I mean by that is that A is always going to pair up with T. So A is complementary to T. C is always going to match up with G. So C is complementary to G. So you can see now that if we took the 2 strands and separated them and looked at just one strand, we could figure out the sequence of the other strand. It would be complementary to the first strand. The 2 strands are also said to be anti-parallel, and that refers to the fact that the 2 strands are oriented in opposite directions. Once again, that relates back to the way that they have to be built. New nucleotides have to be added to a 3' hydroxyl. And that means that our sugars are going to be oriented in opposite directions. In any molecule of DNA, no matter how short or how long, we're going to have 2 strands, and each strand is going to have a 5' end and a 3' end. Those numbers, 5' and 3', refer to the carbons on the deoxyribose. So at the 3' end, we have this exposed hydroxyl, and that can engage in a dehydration synthesis reaction. And this is the only place that a nucleotide can be added. Nucleotides cannot be added to the 5' end. So as the cell is building DNA and building new strands, it can only extend the 3' end. To sum up, RNA and DNA are similar when it comes to their components, but they're a bit different in the way that they're built. So first of all, in RNA, the sugar molecule is slightly different. It has an extra oxygen. Or another way to think about that is that DNA is missing an oxygen. RNA is single-stranded. We have 1 helix. That makes RNA a rather delicate molecule. There are lots of enzymes in the cell and on the surface of your body, etc., that will gobble up RNA and break it quite easily. Finally, remember that RNA contains uracil in place of thymine. When it comes to naming the bits and pieces that make up nucleic acids, the terminology can get a bit overwhelming. And we don't need to get into all the details, but I'd like to point out the difference between nucleoside and nucleotide. And that's because you're likely to run into both of these terms while you're studying. I've been using the term nucleotide, and that is a sugar, a base, and one or more phosphates. So we can have nucleotides that have one phosphate, two phosphates, or three phosphates. ATP, our favorite molecule, adenosine triphosphate, is a nucleotide. It can be used to shuttle energy around, but it can also be used to build RNA. A nucleoside refers to just the sugar and the base. And you might be wondering, well, why bother? Why do we have these differences? Well, when we're talking about an intact DNA molecule, for instance, we don't really care about the phosphates. The information is contained within the sequence of nitrogenous bases. And the phosphates are just there to hold them together. They're there to form part of that phosphodiester linkage. So quite often people will ignore that part of the molecule. Incidentally, on this set of diagrams here, the first one, of course, is a nucleoside. We don't have a phosphate. The second two are nucleotides with a T, but note that the one on the right, the far right, is a deoxynucleotide. It's missing an oxygen at the second carbon as indicated by the arrow. The one in the middle is an RNA nucleotide. DNA consists of two strands of nucleotides twisted around each other. Think of it like a spiral staircase, and the rungs of that staircase are the pairs of nucleotides. The pairs of nucleotides are held together by hydrogen bonds, and hydrogen bonds individually are quite weak and fairly easy to break. There's a lot of them, so that maintains the stability of this molecule, but it's not that difficult for enzymes to unzip a few of those nucleotides at a time. The DNA molecule acts like a zipper. We can unzip it and we can use each strand as a template to make another strand or simply to transcribe the information that's contained within the molecule. Each strand of nucleotides within a DNA molecule can serve as a template. The reason for that is the complementary nature of nucleotides. What you're seeing here is a replication fork. We have a DNA molecule that's splitting into two strands, and the exposed nucleotides are attracting, through hydrogen bonding, their complementary nucleotide. You can see at the bottom that as these new nucleotides connect to each other, they form complete new DNA molecules. As Watson and Crick were deciphering the structure of DNA, it became apparent to them that DNA was replicated by this simple technique, whereby the two strands were separated and each served as templates. In their 1953 article, they stated, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." So what you're seeing in this diagram is a parent DNA molecule, shown in dark blue, that splits into two strands, and that's accomplished by breaking those hydrogen bonds. Now that we have individual strands, they can attract complementary nucleotides that then bind together by dehydration synthesis to give us these new strands shown in light blue, and we end up with two daughter DNA molecules. Recall that DNA in bacteria always occurs in a circular form. We have one big chromosome, and it's one big loop of DNA in the center of the cell. We can have plasmids as well. Plasmids are tiny little circles that only contain one or a few genes, and we can have multiple copies of plasmids within a bacterial cell. On the chromosome, and also on the plasmids, we have a sequence of DNA known as the origin of replication, sometimes just shortened to ORI. There are proteins that recognize this specific sequence. They bind to it and they allow an enzyme known as DNA polymerase to bind at that spot. DNA polymerase, as the name suggests, is an enzyme that builds DNA. It takes monomers, nucleotides, and links them together to make a polymer, to make a new strand of DNA. The DNA is unzipped into two strands at the origin of replication, and then DNA polymerase can do its work. It's going to build new strands, daughter strands, using the parental strands as templates. That area that was opened up as the DNA was unzipped at the origin of replication is known as a replication bubble. At either ends of the replication bubble, we have replication forks. This is where the parental DNA is being unzipped a few nucleotides at a time. DNA polymerase is progressing in both directions, expanding that replication bubble. And eventually those replication forks will make their way in opposite directions all the way around the plasmid and eventually meet. The image that you're seeing at the bottom here is a scanning electron micrograph. It's showing a plasmid in the process of replicating. The arrows are pointing out replication forks. They're moving towards each other and they will eventually meet. And of course, when they do, we will have two separate loops of DNA. There are enzymes that will seal those daughter strands together. And that's what you're seeing in the diagram. Note that the two daughter DNA molecules consist of one parent or template strand and then one brand new strand that was built from that. Bacteria love their circles. Whether we're talking about the chromosome itself or plasmids, we have circles of DNA that have that origin of replication where the two strands will split so that copying can occur. We can take advantage of this and we can be rather tricky. We can create artificial plasmids. We can take a piece of DNA and it can contain whatever sequences we want. We can bend that into a circle and then add in an origin of replication. If we do that and then we insert that into a bacterial cell, that bacteria will blindly copy it. Because so long as there's that origin of replication and the DNA is in a circle, it will be copied. As we'll see, we can even trick the bacteria into decoding any genes that we put on that circle and expressing those genes, decoding them and using them to make proteins. We've been talking about DNA replication and we focused on bacteria. And that's because things are a bit easier in bacteria. We have that one great big circle of DNA. In eukaryotes, we have lots of individual chromosomes and they're straight or linear pieces. And that complicates things. We'll come back to that a little later in the course. But right now, I want to talk about how the information that's in DNA is used by a cell. Remember, there's two steps, and the first step is known as transcription. Think about what that word means. If you're transcribing something, you're making a copy of it. So maybe you're listening to me right now and you're copying down what I say in your notes. You would be transcribing my words into your notebook. Or maybe you're reading the closed captioning that's on this PowerPoint. Well, that closed captioning would be referred to as a transcript. So you haven't changed the information and you haven't changed the language of the information either. Now, I like to think about DNA and RNA as a language, and the alphabet is nucleotides. So we have these 4 nucleotides in DNA and 4 nucleotides in RNA, and that makes up a language, a 4-letter language. What happens during transcription is that the DNA is going to be pulled apart into 2 strands. Remember again that the hydrogen bonds are pretty easy to break. Individually, they're pretty weak, and your cells can pull apart the strands just over a short distance to have access to that information. There's an enzyme, a very large molecule known as RNA polymerase, and it will build RNA out of RNA nucleotides. And that's what you're seeing happening in this diagram. So we have our DNA that's being pulled apart into two strands. We have a template strand that's going to serve as the template for our RNA transcript. That is the copy of the information that's being made by RNA polymerase. And then we have another strand that's referred to as the non-template strand. Now notice in this diagram it also refers to those strands as the antisense strand and the sense strand. We'll get back to what that means when we talk about viruses. But the DNA molecule is going to be unzipped, and one strand, the template strand, is going to be transcribed into an RNA transcript, more commonly known as a messenger RNA. And here we're seeing that happening. So RNA polymerase is a large, complex structure. It's made up of several subunits of protein. There's a groove that the DNA runs along, and as it runs through that groove, the DNA is split into the template strand and the non-template strand. The template strand is read by RNA polymerase, and the appropriate complementary RNA nucleotide is going to be added. As they come in, they join to the one that was added previously. And then our newly made messenger RNA or RNA transcript will leave through a little tunnel in this molecule. So you can see RNA polymerase is moving along the DNA molecule, opening it up a little bit at a time, and it will re-zip as the RNA polymerase passes. And as it opens it up, it reads that template strand, forms a messenger RNA, and then the messenger RNA gets spit out the back of RNA polymerase. And here we're seeing the same thing again, just in a bit more detail. So the different colors that you see here are different large proteins that make up this very complex enzyme. Let's take a look at a hypothetical example. That sequence that I've written in red represents a hypothetical template strand sequence from the gene of a chromosome. What's going to happen is RNA polymerase is going to come along, and it's going to move in this direction. And as it does that, it's going to read the information on that template strand and use that to make a complementary messenger RNA strand, a transcript. So the first letter it sees there is a T. Well, the complementary nucleotide would be an A. The next nucleotide that it sees in the DNA is an A. Well, if we were talking about DNA, then the complementary nucleotide would be a T, of course, but we're talking about RNA, so it's going to insert a U and it's going to attach it to that A. Then it sees a C, the complementary nucleotide is a G, it's going to be added to that growing chain, etc. And this will proceed down the entire length of the gene, which of course would be much larger than what you see here. A gene codes for one messenger RNA, and that messenger RNA codes for one protein. Now, to be honest, it's a little trickier than that because the messenger RNA can be modified before it gets to the ribosome. So we can actually have a gene coding for proteins that are tweaked a bit, so similar forms of a protein. But for our purposes, we're not going to worry about that. That's a bit beyond this course. A gene is defined by two sequences. There is a promoter sequence at the start of the gene, and there's a terminator sequence at the end of the gene. So these are sequences on the DNA. And they tell the RNA polymerase where to start and where to stop. So RNA polymerase will line itself up at the promoter. It'll start unzipping the DNA. It'll move to the terminator, and when it gets to the terminator sequence, it will stop and detach from the DNA molecule. The messenger RNA is released, and of course that's going to travel out to the cytosol where translation will begin. Translation is the second step in this process of using the information within DNA. And again, think about what the word means. Translation is a change in language. We started with nucleic acid, DNA, and then RNA, and that consists of a language with an alphabet of 4 letters, 4 nucleotides. Now what we're going to do is we're going to go from the language of nucleic acid to the language of protein. Protein has an alphabet of 20 letters, 20 amino acids. So we have 20 different amino acids that need to be coded for. How is that done? Well, it's done using codons. A codon is a sequence of 3 nucleotides on the messenger RNA molecule. All living things use the same genetic code, and that's because all living things can be traced back to the first population of cells that appeared on the Earth, and they were the ones that evolved this genetic code. So whether we're talking about humans or penguins or dragonflies or bacteria, it doesn't matter. All of these living organisms use the same code. And that means we can take DNA from one organism and put it into another, and it works. It will work in that organism. It will be decoded into the exact same protein. Viruses use this trick as well. Viruses use the same code. They can take their genetic information, put it into a cell, and trick that cell into decoding it. So let's take a look at a little example here of how this decoder that you see in front of you actually works. So we've got a short little sequence here that consists of only 2 codons. We've got CUA, that's our first codon, and GGC would be our second codon. Let's try to decode that first codon. So our very first letter in that triplet of nucleotides is a C. So we go here, where it says first letter, and we identify the C as our first letter. Our second letter on this axis is a U. And our third letter over on the right is an A here. And then we look for where all of those intersect, and we find the codon we're after right here, CUA. And that codes for the amino acid leucine. We could take a look at our second codon, GGC, and if we follow the same format here, we can find GGC down the bottom right, and that codes for the amino acid glycine. So if you know the sequence of a messenger RNA, you can figure out exactly what protein it codes for. You can figure out the sequence of amino acids that's going to result. Now, one thing to note is that we have 64 different possibilities if we're looking at a triplet using an alphabet that consists of 4 letters. But we don't have 64 different amino acids. We only have 20. And that means there's a lot of redundancy. So if you look at leucine, there's 4 different ways that we can code for leucine. Some of the other ones, though, there's only 2 ways. So what have we got here? Phenylalanine, for instance, there's only 2 ways to code for that. So it depends on the amino acid. We have some special codons as well. We have 3 codons that are stop codons, and they do not code for an amino acid. Instead, when the ribosome gets to that sequence, so a UAA, a UAG, or a UGA, the ribosome stops translating, and the ribosome falls off of the messenger RNA. So this is the sequence that tells the ribosome to stop translating. And then we have one very special codon known as the start codon, AUG. And that is the only codon I want you to remember. If I ask you to decode a sequence, I will give you a decoder, but you should be familiar with that one codon, AUG. Now it does code for an amino acid. It codes for methionine. And for this reason, methionine is always the first amino acid in a protein. So it tells the ribosome where to start translating, and the ribosome does actually read that codon, and it adds a methionine as its very first amino acid. Now, quite often that methionine is going to be cut off later. In many ways, the functioning of a ribosome is similar to the functioning of RNA polymerase. RNA polymerase will travel along one strand of DNA, read the information that it contains, and use that to make a messenger RNA. But remember, this is pretty much straight up copying. The ribosome is going to move along a messenger RNA, read that information, and use that information to make a sequence of amino acids, so to make a protein. And you're seeing that happening in this diagram. So on that diagram, the messenger RNA is shown moving to the left. But we could just as easily think of that as the ribosome moving to the right. It's all relative. Anyway, the ribosome is going to move relative to the messenger RNA, and as it does, it's going to read the codons that are on the messenger RNA. So we have our ribosome. Our ribosome consists of two parts, and they're clamped onto that messenger RNA. We have the messenger RNA itself, and then we also have something called a transfer RNA, or tRNA. And these transfer RNAs are responsible for transferring the appropriate amino acid to the ribosome. So let's take a look at that. We have a transfer RNA here, and you can see that it's bound to glycine. At the other end, we have an anticodon, and the anticodon in this case is CCG. Now, any transfer RNA that has the anticodon CCG will always associate with glycine and nothing else. That's what makes the whole genetic code work. If that wasn't the case, we wouldn't have a genetic code. There are enzymes that look at that anticodon and add the appropriate amino acid to the other end of the transfer RNA. That particular anticodon is complementary to this codon here, GGC. So the anticodon will bind to the codon, and it won't bind anywhere else. So this is how that code is decoded. So what's going to happen is the transfer RNA is going to bring in the appropriate amino acid, and after that happens, everything is going to shift. The ribosome is going to shift to the right, or the messenger RNA to the left, however you want to think about it. And that transfer RNA is going to move to the second slot in the ribosome. The transfer RNA that was already in that second slot is going to get kicked out. The other thing that's going to happen is the glycine is going to be added to this phenylalanine. So it's going to be added before that previous transfer RNA is kicked off of the ribosome. So every time the ribosome moves, we have a new incoming amino acid that's added to the end of the chain. The chain keeps growing until our ribosome gets to a stop codon, and then everything falls apart and the protein is finished. Let's quickly take another look at this process. So on our messenger RNA in this example, we have a start codon, AUG. And that's the sequence that informed the ribosome where it should start. That told the ribosome where to clamp onto the messenger RNA and start decoding. A transfer RNA with the anticodon UAC bound to that codon, and that anticodon is always associated with a transfer RNA that carries methionine. Then we had a second transfer RNA bind to this codon, ACG. And this transfer RNA brought in with it serine. The next thing that happened is the serine was bounded to methionine to form a peptide bond. And as you know, that's a result of a dehydration synthesis. Now at this point, that first transfer RNA can leave. It's contributed its methionine. It'll go off and find another one and wait until it's needed again. So these transfer RNAs are recycled. Now we have a new transfer RNA coming in, and it's recognizing this codon, CAA. So the movement of the ribosome opened up this spot for that new transfer RNA to bind. This transfer RNA is carrying glutamic acid, and now it's going to be transferred to the serine and form a peptide bond. And then once again, as things shift, the second transfer RNA is going to be kicked out and recycled, and a new one carrying proline is going to enter. Transfer RNA is an odd molecule. Now, I've stated that RNA is single-stranded, and that is usually the case. What you see here is a single strand of RNA, but it's bent back on itself, forming kind of a cloverleaf structure. And in some parts, it's held together by hydrogen bonds. So you could almost say that sections like this are double-stranded RNA in a way. At the bottom, we have an anticodon. Very important, it's going to recognize a codon on the messenger RNA. And at the opposite end, we have this special sequence here that will recognize the appropriate amino acid for that anticodon. Again, that job of matching up the appropriate amino acid with the appropriate transfer RNA is done by a group of enzymes. And that job's pretty important. If they get it wrong, the whole genetic code falls apart. Now, one last thing you might notice that you might be wondering about is we have some nucleotides here that don't have letters. Instead, they have a little asterisk. And the reason for that is because I kind of lied about there only being 4 nucleotides in RNA. Transfer RNA has a few special nucleotides, but we won't get into that. Remember back in Biology 111 when we talked about the structure of proteins? Remember how complicated they are and how complicated their folding can be? Well, the sequence of amino acids determines the structure and the shape of a protein, and it determines what that protein can do. What you're seeing here is the assembly of amino acids into a polypeptide or protein. And you're seeing the functional groups highlighted in blue. Remember, that's the part that sticks out from the amino acid. It can be very simple, so we can have an R group or functional group that's just a hydrogen. But some of them are quite complicated. We can have large ring-shaped structures, for instance. We can have some amino acids that have a charge, and that means that they're going to interact with water. They're going to be hydrophilic. They'll interact with other molecules that are charged. We can have other ones that are hydrophobic. That's going to have a big impact on how this protein folds itself and how it interacts with other parts of the cell. We can have some that contain sulfur that can be involved in covalent bonds that will help stabilize the protein, etc. So what it all comes down to when we're talking about what a protein does, it comes down to the sequence of functional groups. And that is ultimately determined by the sequence of nucleotides in the DNA. Back in the day, kids used to get excited about decoder rings. So you could wear this ring and you could change the settings on it and share those settings with your friends, and then you could use it to send and decode secret messages. Now, we've looked at the classic table that you see in most textbooks that's used to decode messenger RNA sequences. I kind of like this decoder better, and I usually refer to it as a decoder ring. If I were to give you a question on a test, I'd probably give you this decoder ring to solve a sequence that I might present you with. So briefly, I'll just show you how you use this. Let's say that we had the codon GCA. Your first letter is in the middle. So we start with our G in the center of the decoder ring. Our next letter is a C. So that's in the second spot, starting from the middle. And then our third letter is an A. And that codes for alanine. That would be the amino acid. So I really like this ring. I think it's much easier to use. Now, one thing I should point out, I mentioned that the genetic code is redundant. Well, notice that for a lot of the amino acids, like alanine, the last letter in the codon, so the nucleotide in the third position, doesn't matter. It could have been a G, an A, a C, or a U. We still would have had alanine. That last codon is sometimes referred to as the wobble position because it really doesn't matter what's there. Not for all of the amino acids, but for quite a few of them. Let's take a look at a more complex example. Let's take a look at this hypothetical messenger RNA. Now, this messenger RNA is much, much shorter than a real messenger RNA, of course. And the other thing to note is that in a real messenger RNA, there would be nucleotides preceding the start codon, and there'd be nucleotides after the stop codon. The ribosome simply wouldn't decode those nucleotides. The ribosome is going to look for the AUG codon. That's the start codon, and this is how it knows where to begin. It's also how the ribosome knows how to divide this message up into the appropriate three-letter words. Let's just imagine that the ribosome didn't bother looking for an AUG. Let's say it didn't have to. It could start wherever it wanted. Let's say that it decided to start with UGA. Well, if it did that, every single codon would be incorrect. We would get an entirely different protein. So the AUG lines up the ribosome and sets up the reading frame. Now this messenger RNA is going to be used to make a protein, of course, a sequence of amino acids. So we have AUG coding for methionine. That's always going to be the case, but as I mentioned, the methionine might be removed later. Then we have ACU coding for threonine, AGA coding for arginine, GGA coding for glycine. Now the very last codon, UAA, does not code for an amino acid. Instead, it simply tells the ribosome to fall off of the messenger RNA and stop what it's doing. Now also note that the protein has an N terminus and a C terminus, an amino terminus and a carboxyl terminus. The protein has a direction to it. It has a polarity. Now let's have a look at the applications of molecular biology. Again, the genetic code is universal, and that means we can take DNA from one source, one organism, and put it into another organism, and it will function in that second organism. If we're taking two different bits of DNA and we're cutting them and pasting them together, we call the resultant DNA recombinant DNA because we've recombined things. What you're seeing on the left is a box of laundry detergent. And this particular laundry detergent contains lipase, which breaks down lipids, and protease, or proteinase, which breaks down protein. That means that the laundry detergent will do a better job. Now, what researchers did is they found the genes for these enzymes, isolated them from bacteria, isolated the resulting proteins, the enzymes themselves, and figured out ways to make those enzymes function better. So they found if they, change the sequence slightly, maybe they could get a more effective enzyme. They tweaked the genes so that they would code for these new and improved enzymes, and then they put those genes back into bacteria. And the bacteria will express these genes. What I mean by that is they will use them to make protein. They will use them to make these new and improved enzymes that can be put into your laundry detergent. On the right, you're seeing something called humulin, which is insulin. It's human insulin that was produced by bacteria. So they've taken the gene for human insulin from humans, put that into a plasmid, put that into bacteria, and trick the bacteria into making human insulin that can then be collected quite easily. Note that on the box there, it says that this is human insulin of rDNA origin. That R stands for recombinant. When you take genes from two different organisms and bring them together, we say that you've created a transgenic organism. And you can do some rather scary things with this technology and also some rather wonderful things. Now what you're seeing here in A is a tobacco plant that can actually express a firefly gene. The gene that would make a firefly's butt light up has been put into this tobacco plant and it can light up now. The pig that you see here contains a gene that codes for a fluorescent protein that's found in jellyfish. And it's being turned on just in certain parts of the body. So you can see that his toes, his nose, his extremities are lighting up, or more correctly, fluorescing because of this jellyfish gene. And you might think, why in the world would someone do something like this? Well, these turned out to be very, very useful research tools. What you can do, for instance, is you can take that jellyfish gene, it's something called green fluorescent protein, GFP, and you can attach it to a promoter, an on-off switch that you want to study. So for instance, let's say you have a promoter, but you don't know what cells turn it on or what cells turn it off. Well, you could take that promoter, you could attach it to your jellyfish gene, you could put it back into the animal you're studying, a lab rat for instance, and you could find out what cells use that promoter. You could use it with cancer cells. If you want to know if cancer cells turn on certain promoters, well, you could attach the promoter to a jellyfish gene and see what cells light up, or see when cells light up during development. So there's all sorts of really important research that can be done with these tools. In order to form recombinant DNA, we have to cut DNA and then splice it together. The cutting is done with a group of enzymes known as restriction endonucleases. These are enzymes we've known about since the 1970s, and we've isolated thousands of them. And what's really fascinating is these enzymes all cut at very specific sites. So if we know the sequence of DNA that we want to cut, we can find an enzyme that will cut at that exact spot. Incidentally, these are enzymes that were isolated from bacteria. They're used by bacteria to protect themselves from bacteriophages. Bacteriophages are viruses that inject their own DNA into bacterial cells. And the bacteria use these enzymes to chop up that DNA and try to thwart the advances of these bacteriophages. So you're seeing two examples here. At the top, we have an enzyme known as ALU1, and you can see that it recognizes the sequence AGCT. And I'm reading that in the 5' to 3' direction. Now if we look at the bottom strand and read in the same direction, 5' to 3', we have the exact same sequence, AGCT. So these sequences are palindromes. A palindrome is something like the word race car. It's the same forwards and backwards. The arrows show where this particular enzyme will cut. It will cut between the G and C on the top and bottom strand. Now that's going to give us a blunt end. And what I mean by that is we don't have any single-stranded bits of DNA hanging over. Now compare that to Echo R1. And I should mention these enzymes are named after the organisms they were first discovered in. So Echo R1 came from E. coli. So on the bottom there with Echo R1, we have this sequence GAATTC. And we have the same sequence on the bottom strand, again running in the same direction. We have cuts being made between the G and A. So on both the top and bottom strand, we're going to get a cut here and here. And the hydrogen bonds holding the rest of that sequence together are also going to be broken. And that gives us what's known as a sticky end. There's a single strand overhang or underhang on each of the resulting molecules. If those sticky ends come together, they will naturally hydrogen bond. And then we can add another enzyme known as ligase that will seal up the gaps and seal things back together. So let's take a look at how we might go about that. At the top, we have part of a sequence of DNA. And imagine that this is a plasmid. And we're just zoomed in on that restriction site. That's where echo R1 is going to cut. And when it cuts, it's going to create these sticky ends. Well, now we can take a fragment of DNA that we're interested in from something else. It could be from a different species. And we're going to cut it with echo R1 as well to produce the same sticky ends. Now when we take these bits of DNA and we put them together in a test tube, the DNA will come together to form the most stable configuration, which is the configuration in which the highest number of hydrogen bonds can be formed. So that cut piece will slot itself into the cut that we made in our plasmid. Then we add an enzyme known as DNA ligase, and what DNA ligase is going to do is it's going to reform the phosphodiester bonds here and here and here and here. So basically it's going to act like glue, and now we have our recombinant DNA molecule. So we can take a plasmid from bacteria, cut it with echo R1, and then take a piece of DNA out of the chromosome of a eukaryotic cell, so it could be the human insulin gene, for instance, cut the ends of that gene with echo R1, bring them together, ligate them or glue them together, and then take that and put it back into a bacteria cell, and now that bacteria cell will carry on as if that gene was meant to be there. It will make copies of this, so every time the cell divides, it will copy that DNA. And also, if it's put in correctly, it will express that gene. It will use it to make protein. Some amazing things have been done with this sort of technology. So let's say you're studying development in humans, and you're interested in the role played by a particular protein. But it's not a protein that's found in great quantities. Maybe it's only found in an embryo. But you want some of that protein to study. Well, you could take the gene for that, and you could put it into bacteria or maybe into yeast cells, and they could grow up a whole lot of it for you that you could then use in your research. Or we could use it to engineer plants that are resistant to pesticides. We could use it to engineer bacteria that are able to clean up oil spills. We could use it to make pharmaceuticals. We can use it to make hormones. So the hormone might be something like human growth hormone, like you see here. So we can grow up enough of that that we can give that to a child that's not generating enough on their own. There are many companies that sell commercial plasmids, plasmids that have been tweaked and engineered from scratch that you can use in your research. And I should mention the E. coli that are used in this sort of research are crippled. They've intentionally been mutated so that they're missing something from their chromosome, and you need to add something typically to their diet that they can't make on their own. So that means they can't survive outside of a lab. This particular plasmid here has a gene for ampicillin resistance. Ampicillin is similar to penicillin. And you might be wondering, well, why would you want to have that on a plasmid? You give this plasmid to a bacteria, and now it's resistant to ampicillin. Well, if you're working in a lab and you're growing up a lot of bacteria, there's a good chance that some of your Petri plates might become contaminated. But if you add ampicillin to your agar, then the only thing that can grow on the plates is a bacterium that has this resistance gene. It makes things a bit easier. You know that any bacteria growing on your plates are bacteria that contain the plasmid you added. This plasmid also contains a gene called the LAC-Z gene, and this codes for an enzyme that can break down a substrate and change the color of the cell. So if you add something called beta-galactose, type of sugar, to your plates, and the cells have an intact LAC-Z gene, then they will change color. They will become blue. The colonies will become blue. Now, embedded within this LAC-Z gene is something called a multiple cloning site. And down the bottom, you're seeing a zoomed in area of that multiple cloning site. All of those words in blue are different restriction endonucleases. And you can see the one we talked about previously over here, echo R1. So you can cut a piece of DNA that you want to amplify or express with echo R1, for instance, and then cut your multiple cloning site with echo R1 and then stick your piece of DNA in there. Now what this does is it will interrupt that LAC-Z gene. If you put a great big chunk of DNA in there, that LAC-Z gene no longer works, and the colonies won't be blue. So this tells you a lot, and it helps you sort through hundreds of plates very quickly and identify the colonies that you're interested in. Remember that when you're spreading bacteria on a Petri plate, each colony that results came from a single bacterium, and every single bacterial cell in that colony is genetically identical for that reason. So if you look at your plates, first of all, you know that anything that grew on the plates has your plasmid, because it has ampicillin resistance. If you look at the color of the colonies, anything that's white instead of blue also has an insert if you inserted something into the multiple cloning site. Now, I just went through a whole lot of stuff here that maybe went over your head, and that's okay. I just want you to realize how easy it is to manipulate DNA and how powerful this technology is. And the stuff I'm talking about right now is quite old, actually. We've been able to do this for decades. There's more powerful technology out there. So we can stick DNA into a plasmid, stick that into a bacterium, and trick the bacteria into making lots of copies of this DNA for us. But there's an easier and faster way, something called the polymerase chain reaction. And this is something that's done without any cells whatsoever. It's done in tiny little test tubes in a machine called a thermocycler. And all this machine does is heat up and cool down really quickly. Every time it heats up, the DNA splits. It separates into two strands. It cools down a bit, and the enzymes and other materials that are included within the tube allow us to make copies of our target sample, our DNA of interest. The polymerase chain reaction was perfected and marketed by a man known as Carey Mullis, who unfortunately passed away a couple years ago. But he was a rather interesting fellow, to say the least. He was a Nobel Prize winner in biochemistry, he was a surfer, and he had some interesting tales about talking to fluorescent extraterrestrial raccoons. He may or may not have been on psychedelic drugs at the time. Anyway, what you do with the polymerase chain reaction is you get yourself a tiny little tube, and into that tube you stick the DNA that you want to amplify or make copies of. And perhaps you only want a specific sequence out of a mixture of DNA. So let's say you're doing a forensics analysis. You could take a tiny sample of blood, put it into your tube, but you're just going to copy one little sequence of DNA, maybe one gene, out of that mixture of bits and pieces of DNA. Then we have little bits of single-stranded DNA known as primers. The primers are complementary to the ends of the DNA sequence that you want to copy. We also have DNTPs. These are deoxyribose nucleotides, so A's and T's and C's and G's. And then finally, and this is the really clever part, you include DNA polymerase, but not just any DNA polymerase. You include polymerase from Thermus aquaticus, which is a species of archaea. Remember that archaea are prokaryotes. They're kind of like bacteria, but they live in very extreme environments. And this particular archaean lives in very, very hot water. It lives in hot springs that can be boiling. Now, the importance of this is that its DNA polymerase doesn't denature the way you might expect an enzyme to when you heat it up to boiling. Instead, it remains intact. And that's rather important because in this machine, this thermocycler, we're going to heat up the DNA close to boiling and then cool that down. And we're going to do this over and over and over again. And if we didn't have this special DNA polymerase, well, the enzyme would be destroyed every time. We couldn't do this through several cycles. Okay, so once you've mixed all this stuff in your tube, you start the machine through its cycling process. What it does first is it denatures the DNA. Now, when I've used the word denature before, I've used it in reference to proteins. Unraveling a protein is denaturation. We can denature DNA as well, but it means something a bit different. It means that the DNA is going to split into two strands. It's also referred to as melting. So we're going to heat up the DNA for 30 seconds, and the DNA is going to split into two strands. Then we cool it down a bit for about 30 seconds again, and this is a period known as annealing. And it gets just cool enough that hydrogen bonds can form, and that allows our primers to bind to the ends of our target DNA, the sequence that we want to copy. Then we keep it at about 72 degrees for 45 seconds, and during this time, DNA polymerase is going to build off of the 3' end of our primers, and it's going to copy our target DNA. It's going to use the target DNA as a template. And then this cycle is going to happen over and over and over again, and every cycle will double the amount of target DNA that we have in our tube. PCR is a very powerful procedure for two reasons. First of all, we don't need very much material to start with. And secondly, that material that we're interested in can be mixed in with a whole lot of material that we're not interested in. So imagine that we want to isolate and make copies of one human gene, and we have just a small number of human cells in a test tube. We can amplify or copy that one target sequence even though it's mixed in with tens of thousands of other genes and a whole lot of junk DNA. So we can isolate that one gene from the entire genome, which is the complete set of genes that make up an organism. So here's how that happens. We have our DNA in a tube, and along with that we have nucleotides, and we have DNA primers that are complementary to known sequences of DNA at the end of our target. Now we also have a few other things. We have some buffers, we have magnesium chloride and a few things like that. And then we have our DNA polymerase. So what happens is we heat up this machine, we heat up the water in the tube, nearly boiling, and that causes the DNA to split, to denature. And now we have these two exposed single strands of DNA. The next thing that happens is the DNA primers will bind to opposite ends of that target sequence. So we have two primers that we've designed. It's pretty easy to make a primer. You just figure out what the sequence is. You send that sequence off to a company, they make a primer for you, and they send it back to you. So these primers are complementary to sequences at the end of the target. Now this is not to scale. The DNA primer is going to be less than 40 nucleotides in length, and usually the piece of DNA that you're copying is hundreds or thousands of nucleotides. But during the annealing period, we cool things down just enough so that hydrogen bonds can start to form again. So that denaturation process broke the hydrogen bonds. But now we're allowing hydrogen bonds to form again, but the DNA isn't zipping back up just yet. Instead, our primers are binding to their complementary sequences. The next thing that happens is the extension period, and this is where the DNA polymerase is going to be allowed to grab onto those nucleotides that are floating around in the tube and build off of those primers. Now that's not something that I've talked about yet, but DNA polymerase has to build off of something. It has to build off of a primer, and in cells, when DNA replication occurs, it's a similar process. Your cells have primers that DNA polymerase has to build off of. Now, one thing to note is if we look at the primes, the ends of these molecules, With our primer, we have a 3' here and a 5' here. Remember that DNA molecules are anti-parallel. We have a 5' here and we have a 3' over here. And this dictates which way the template is going to be used. This can only be built in this direction. This up here can only be built in this direction. And in that last diagram there that shows the extension, that light blue strand of DNA is brand new DNA. This cycle of denaturation, annealing, and extension is going to happen over and over and over again. And because our primers bind to sequences at the ends of the target DNA, it's the target DNA that's going to be copied. After one cycle, we've doubled the amount of DNA. After 2 cycles, we've quadrupled it. After 3 cycles, we have 8 times as much target DNA. And after 30 cycles, you can see we have 10 to the 9th copies of our target DNA, and it's going to far outnumber the original DNA that we started with. So the junk that we started with is going to be such a tiny fraction of what's in the tube. The tube is going to be almost pure target DNA. One more technique that's essential to molecular biology is gel electrophoresis. What you're seeing in this photograph here is an agarose gel. So it's this dense gelatin-like material that's made out of something called agarose. And within this gel, there are wells, little depressions, and you can see them here. Now what's being done is someone has taken some of the DNA they're interested in, and they've mixed it with a dye, so they can see what they're doing as they load this thing, and also mixed it with some glycerol, which will allow the DNA to sink to the bottom of the well, because this gel is submerged. It's submerged in a buffer that will conduct electricity. DNA has a slightly negative charge, and that's because it's got negative phosphates that stick out on the outside of the backbone. If you run an electric current through this apparatus, the DNA will be repelled from the negative terminal and move towards the positive terminal. Now DNA has to kind of snake its way through this dense gel. The larger molecules are going to be impeded by it more than the smaller molecules. So if you let this run for an hour, the really big fragments of DNA won't get very far, but the smaller ones will. So the DNA is going to separate out by size. Now there are dyes that you can add to the gel afterwards that will bind to the DNA and let you see where it is. Some of the old school dyes were fluorescent dyes, and when you expose them to UV light, they would light up. Incidentally, we have this equipment and we have a PCR machine in the lab, so you will have a chance to see this, and hopefully this will make a bit more sense to you then. Let's put this all together by looking at an example. Imagine that you have a patient that you suspect might have chlamydia. How are you going to test for that? Well, you might think that maybe you could culture chlamydia. Well, the problem is chlamydia is an obligate intracellular parasite. It can only reproduce inside a cell. So you can't just culture it on a petri plate. It's not going to survive. You could perhaps do cell culture, but that's a lot of work, and there's an easier way to do it. What you could do is you could look for the presence of chlamydia-specific DNA sequences. So this patient is going to be shedding epithelial cells into their urine. You can take a small sample of that urine and use PCR to find out if there's any chlamydia DNA, which there would be if he has chlamydia living in those shed epithelial cells. So let's imagine that we're looking at just that little bit of DNA that you're interested in. So this is part of a great big circle of DNA. So remember, even though chlamydia are quite simple, they are still bacteria. They do have a circular chromosome. And we are interested just in this little bit from here to here. So just a tiny fragment of the entire circle of DNA. And we're looking at double-stranded DNA here. Well, what you can do is you can design primers that will recognize the ends of your target sequence. So you design a primer that you know will bind to a DNA sequence that's specific to chlamydia. You've got a primer at one end, you've got a primer at the other end. And then with PCR, what's going to happen is those areas are going to be amplified. So we're going to make copies just of that target sequence. Now let's say that we know the size of that. Chlamydia is being sequenced, as have a lot of other organisms, and even if they haven't been totally sequenced, we know about several genes that are present and specific. So let's say that we know this gene is 260 base pairs in length. Now what do I mean by base pair? A base pair consists of two nucleotides bound together, so one on each strand. So we have 260 nucleotides between those two purple lines I've drawn. Well, now what we can do is we can do our PCR, and then we can take the sample that we get out of the PCR reaction and run it onto an agarose gel, and we can look to see if we have a fragment of DNA that's 260 base pairs in length. That's what you're seeing on the gel here. The very first lane that's marked M contains marker DNA. It contains bits and pieces of DNA of known sizes that you can compare your target sequence to. We have some lanes here, two that contain our PCR'd sample of urine and then a negative control from someone we know doesn't have chlamydia, and we can compare them. And sure enough, note that in our patient here, we have this 260 base pair, very noticeable band. My arrow is not noticeable, but very noticeable band, and my arrow is still not noticeable, but very noticeable band here that tells us that chlamydia is indeed present. So this is a very quick and easy way to find out if a certain pathogen is present, and it's something that would only take a few hours to do. Okay, that was a rather intense topic. Quite a lot of technical stuff. Make sure that it makes sense to you before we move on to viruses. Make sure that you understand the central dogma of molecular biology, and you understand the role that DNA, protein, and messenger RNA play in cells. Viruses, as we'll see, are just bags of genetic material. They inject their genetic material into cells, and they trick the cells into copying it and decoding it to make protein. To summarize, we have DNA that is used as a repository of information that can be passed down from one generation to the next. The DNA is made up of genes, and genes ultimately code for protein. When we're talking about the entire set of information that's found within a cell, we're referring to its genome. When we're talking about whether or not certain alleles of a gene are present, we're talking about the genotype of that cell. Genotype is going to ultimately determine phenotype. Phenotype is determined directly by proteins. Proteins do all the work in a cell. But the phenotype, which is the outward appearance or behavior of an organism, goes back to the actual sequence of nucleotides in the DNA. We have a messenger that transmits the information from DNA to the ribosome, and that is messenger RNA. And here are our concepts. To summarize, DNA is involved in long-term storage of information in cells. Know how DNA differs from RNA. Know their components and know their roles within the cell. Remember that DNA can be unzipped or denatured because we can break these hydrogen bonds a few at a time without too much trouble. And DNA has to be replicated before cell division occurs. The central dogma of molecular biology states that DNA is used as a template to make messenger RNA. Messenger RNA is going to be read by a ribosome to make proteins. DNA is copied by DNA polymerase, and messenger RNA is transcribed from DNA by RNA polymerase. Messenger RNA contains codons. Codons are triplets of nucleotides that code for specific amino acids. Codons are recognized by anticodons, which are part of a transfer RNA. Transfer RNAs bring in the appropriate amino acid to the protein that's being built by the ribosome. The first codon that's read by the ribosome is AUG, and it codes for methionine. There are 3 stop codons that tell the ribosome when to stop translation. The genetic code is degenerate, which simply means that many of the amino acids can be coded for by more than one codon. The genetic code is universal. All living things share this same code. Genes can be inserted into plasmids that can be inserted into bacteria, and we can trick the bacteria into making copies of that material and even transcribing it and translating it. Target sequences of DNA can be very rapidly copied or amplified using the polymerase chain reaction. PCR can be used to rapidly identify the presence of all sorts of pathogens, including COVID-19, and we will come back to that. And finally, here's our terminology.